Good afternoon, everyone. I welcome you to the talk series CS Katha Barta, where we invite academics and professionals to deliver their talks. Our guest for today's talk is Dr. Chirag Jain. Dr. Jain is Assistant Professor in the Department of Computational and Data Sciences at IISc Bangalore. He leads the ATCG research group that develops algorithms theory and software tools for data-intensive problems in molecular biology. Prior to joining IISc, he was a postdoctoral fellow at National Institutes of Health. He completed his PhD in 2019 at Georgia Tech. Dr. Jain is a recipient of several awards and honors, including DBT Wellcome Trust India Alliance Fellowship, IIT Delhi Graduates of the Gold Award, and NIH Directors Award, to name a few. Today, he will be speaking about algorithms for assembling human genome sequences. I welcome you, Dr. Jain, and request you to take the virtual stage and start your presentation.
Okay, yeah, thank you. So I think you have a very nice initiative at your institute. It's a pleasure to be part of that initiative. And yeah, so I'll talk about problems in computational biology that we work on in my group. And I would actually appreciate questions during the talk. So please feel free to interrupt me. That will give me an opportunity to take some pause and maybe drink some water. So, so yeah, so feel free to interrupt me during the talk. So in this talk, I'll talk about algorithms for assembling human genome, or in other words, uh, algorithms for genome assembly problem. So let me start with some background because I assume many of you are not from this computational biology background. So I'll keep my talk broad and I'll start from basics. So as to start with, let me just tell about the human genome sequence itself. So I think many of you may have seen in your 11th or 12th grades that so we have human genome in our DNA. So the DNA complete DNA sequence is called genome, and we inherit. So we have 23 pairs of sequences, and so each. So if you can see my mouse, so basically we have 23 pairs of sequences, and one. So if you see there are two, for example, two copies of chromosome one, two copies of chromosome two, two copies of chromosome three, and so on. So one copy we inherit from mother, and one copy I would inherit from my father. And we, even though we call them as copies, but they are near identical sequences because the genome of my mother would have small differences compared to genome of my father. So these two copies of every chromosome would be 99.9% identical on average, right? And if we do a sum of all the sequences, that is sum of all 46 sequences, then the entire length of human genome is 6 billion characters. So if I add up all the ATCG characters in all these 46 sequences, then the total length of human genome is 6 billion characters. Okay, so now another background slide is on whole genome sequencing. So when we sequence genome of any individual, we would take their, let's say, blood sample, we would use their sequencing machine. And what we would like is 46 sequences, one for each chromosome. But in actually, because of the technological limitations, we don't get that ideal output. What we get is large number of short fragments or strings from the genome. So these strings would be randomly read by the instrument from random locations of the genome. And what you would have is a large collection of short fragments, which I will refer to as reads during my entire talk. So you have these large number of these reads available to you. So in my toy example, here I'm showing you these reads of length 4, but in practice, the length of these reads will vary depending on the technology being used. So with the latest technologies, the average length of each read is about 10,000 characters. Although I should mention that in all my toy examples, I'll be drawing these short reads only because drawing reads of length 10,000 is inconvenient. So, but just to keep in mind that the average length of each fragment or average length of each read is about 10,000 characters. So now the problem of genome assembly is basically to reconstruct the original genome from these short fragments uh, available to us. So this is a computational problem now. That is, this is the observation that you have that is given by the sequencing instrument. And for many biological applications, we want to know what is a complete genome sequence of that individual. So as a history of this problem, like when the first human genome sequence, like if you talk about the first human genome sequence that was published, so this was a result of the Human Genome Project that was launched in 1990, and the first draft sequence was published in 2001. So this was, of course, a landmark publication, and till date, this sequence is used heavily across all biomedical applications. Now, if you look at the draft sequence that was published in around 2001, so if I just show you a visualization, okay. So before that, let me talk a little bit about how was this process done. So you have this short fragment or reads. So how would you first of all assemble the genome? So to see this picture, let me just draw an analogy with the jigsaw puzzle, which is a common method to communicate or convey what genome assembly problem is. So thinking of genome assembly as jigsaw puzzle, what we can do is each read can be thought as a puzzle piece. And in our problem, we are looking for the genome sequence, but in a jigsaw puzzle, you would want to reconstruct the full picture from those pieces. So similar to how we solve the jigsaw puzzle, that is, we look for the overlap between two pieces. In the genome assembly problem, we would also want to look at the overlap between two reads. That is, a overlap between two reads will likely indicate that the second read appears after the first read in the actual genome sequence. So this overlap information will be an important point to understand how the reads can be ordered to form the complete genome sequence. Now, if we look at the status of the human genome in 2003, it turned out that at that point, only 92% of the complete genome was completed, and many chromosomes had these gaps left. So all the red blocks that you see in each chromosome is indicative of a gap that was left unresolved in each chromosome. And so why these gaps occur? Because the reason these gaps occurred, because these gaps correspond to the repetitive sequences that occur in the genome. Now, why would repeats cause problems? So let me again go back to the jigsaw puzzle to explain, to see why repeats would cause problem. So the reason why genome assembly problem has been challenging for multiple decades is because if you go back to the jigsaw puzzle that we are trying to solve has many repetitive patterns. And if you think of it as if you think of these repetitive patterns, basically now if you have a jigsaw puzzle piece, it will be very hard to figure out where to place it because of these repetitive patterns. So similar patterns in the jigsaw puzzle correspond to repeats in the genome sequence. We are also working with too many pieces because the number of reads that we have to process ranges from typically 5 to 50 million. Also, because the sequencing process is random, the sequencing machine is randomly reading regions of the genome sequence. You will have some regions of the genome where you have large number of fragments, and some regions which are not read enough. So you may have some regions of the genome with few reads and some regions of the genome with large number of reads, for example, because of the randomness. Finally, one other point which is also adds a lot of challenge is that sequencing errors. So the sequencing machines are not guaranteed to read the DNA with 100% accuracy. That is, they will also make mistakes in the reading process. So for example, the true sequence may say A, but they may tell you C. So that these kind of errors also happen in the reading process. And therefore, I can say that in some sense, you can think of it like the pieces are also damaged. So you're solving this challenging jigsaw puzzle with many repetitive patterns, too many pieces, damaged pieces, and so on. And this is the analogy of why genome assembly problem has been challenging and one of the fundamental problems in computational biology. So very recently, yeah, okay. So of course, in the genome assembly problem, you are not given the entire template. So in the jigsaw puzzle, at least you are given the template on the outer board that how should the puzzle final picture look like. But when we are solving genome assembly, you don't know how the final picture is going to look like. So we have to work without any reference in some sense. So very recently, there was a like a milestone achieved in the field, that is the first complete and gapless human genome was assembled in 2022, and this was a result of like lot of progress that was made over the years in terms of the sequencing technology. For example, the sequencing technology got more accurate, the read lengths became longer, and so on. And of course, the improvements in algorithms as well, like the available algorithms to do pattern matching of strings also got faster and more accurate as with time. So as a result of all this progress in the field, a group of researchers in the telomere to telomere consortium published the first complete and gapless human genome assembly in 2022. So now this genome is fully assembled. So the question is, why are we still worried about this problem? So you have this full genome sequence assembled and available to you. So why are we still looking at this problem for research? Well, the main point is that this is assembly of one human genome sequence, but we have 6 billion individuals on planet. So is this solution scalable to those 6 billion individuals? Well, unfortunately, not yet. So the unresolved challenges that are still remaining in the field are first, automation, that significant manual curation is currently required to assemble highly repetitive, challenging regions of the genome. And this manual curation is not scalable. That is, I cannot assemble 1 billion genomes or 1 million genomes because of this extensive manual curation involved for each genome. The second unresolved challenge is also a lack of theoretical foundation. So the current genome assemblers heavily rely on ad hoc heuristics, which don't offer any theoretical guarantees. So this is also a bottleneck, or this has also been an open problem for many years. That is, like the practical solutions that we have for genome assembly heavily rely on heuristics because the input data is so large, and the algorithms that we have that are provably good either make false assumptions or do not scale to that amount of data. The third point I want to mention, which is also unresolved, is that there is significant amount of sequencing data that has to be generated for this complete human genome assembly. And right now, the data generation costs about $4,000 per genome if you want to buy from a commercial vendor. So given these challenges, like there are still, I would say, many open problems in this area. That is, how do you go about addressing all these questions and scaling your genome assembly solution to sequence, let's say, each and every individual on the planet? Okay. So I want to highlight, like, say, we have been working on some of the problems related to genome assembly. So the first point I want to mention is that genome assembly is not a single problem. There are multiple sub problems in this within this topic.
Dr. Chirag, I have one, quick question.
Yes.
Is this a solvable problem? I'm just asking, as in I know what, but I just want to know, is this a solvable problem? Can we do absolutely fast or very cheap?
So you mean solvable in terms of the computational class, like which
Yes.
complexity class it belongs to?
मतलब I don't want like class as a entire community. Do they think that it's a solvable problem and that's why?
So I think one of the bigger challenges is also that there is no clear formulation available that I can say if you solve this formulation, then this problem will be solved. Like, because what I mean to say is that there are a lot of ad-hoc heuristics, and it is an incremental procedure to do step 1, step 2, step 3, step 4. There is no clear problem definition available that I can say this is the input and this is the output that we want.
I
Like,
see.
does that make sense?
Yes, yes, yes.
So there are many attempts made. Like, for example, the first attempt to formulate genome assembly problem was this some problem called shortest common superstring problem. Like, so you are given a set of strings and you want to find out the shortest superstring such that every input string is a substring of that superstring. So this is like a parsimony based formulation, but this and many other formulations turned out to be like, even if you solve them, so even the shortest common superstring is an NP hard problem, but it turns out that even if you have a supercomputer or a quantum computer to solve it exactly, the output will not make biological sense.
Okay.
Because so the like, for example, shortest common superstring is framed under a parsimony assumption that the shortest possible answer is the correct answer, but that's not what, like, biological sequences don't satisfy that property.
Okay, okay. And I believe we will hear from you in the future slides. I was asking like, what is the next step that all this community is looking at? Like, okay, let's solve this part of this entire
Okay.
assembly.
Yeah. Okay. So yeah. So maybe, yeah, I'll like we can discuss that maybe after the talk as well, because so
Yes,
that's like a, yeah, that's like a broader question.
yes,
Yeah.
yes.
Yeah. Okay. Any other questions? Like,
No,
any
I'm
other
good.
questions?
Yeah.
Any other questions from the students? Okay. So let me just, so I want to mention that, so genome assembly right now is not considered as a single problem. Like, one should probably think of it as a, like, a, like, you have to solve probably multiple problems here. So some of the questions that we have addressed in the group are as following. So for example, the first question that we recently addressed is, how many reads do I need to sample from the genome to guarantee the genome reconstruction happens? So this is an information theoretic question. That is, what minimum information do I need during the sequencing process? How many minimum reads should I need to generate? Should be the minimum read length uh, that algorithms require to do unambiguous genome reconstruction under some assumptions. The second question, like, for example, we also recently tackled is how to fix sequencing errors. And so the reason I'm saying genome assembly may be a series of problems is also because that there are multiple steps in this process. So for example, typically most assemblers would start by fixing the sequencing errors in the data. So one can frame an independent problem formulation for just fixing the sequencing errors in the reads before even starting the genome assembly problem. So like this, there are like multiple sub problems in this field that need to be addressed. Suppose now, even if suppose the reads are error free, so the third question that we also recently addressed was, how do you build a sparse graph using suffix prefix overlaps among the reads? And the reason I say sparse is because if the graph is too dense, then it would be hard to figure out the correct walk in the graph that corresponds to the actual genome sequence. So therefore, we would like a sparse chain-like graph that coming out of the like overlap relationships between reads. So in the remainder of the talk, basically what I'm going to do is I'm going to focus only on one topic and try to get into more details of this particular problem. And I'll leave out the remaining for this talk. So as a background, let me describe what an overlap graph means. So let us consider this toy example in which you have a genome sequence. And let me say that you have sampled 5 reads from this genome sequence. So now the overlap graph is defined in the following way, where each read is a node in the graph, and the edges represent suffix-prefix overlap between reads. So for example, you can easily see here that R1 and R2 should have a suffix-prefix overlap. Similarly, R2 and R3 should have a suffix-prefix overlap. R1 here and R5 here don't have a suffix-prefix overlap. So you don't have an edge connecting R1 to R5 directly. So this is a simple definition, like basically each read is a node, and these edges indicate suffix-prefix overlaps between the reads. So now what happens is, like, suppose if you take some similarity data, let's just say in this example, I have taken a small subset of similarity reads from human chromosome 20. Just as a small experiment, and what I did is I created an overlap graph out of a small number of reads from chromosome 20. Now this overlap graph,
Hi.
yeah, go ahead.
Yeah, I had a question. I mean, it's based on the premise of the problem. You said that we don't have an entire picture of what we are looking for, right? That we don't know the final output, like the entire picture of the jigsaw puzzle.
Yes, we don't have the final output. Yes, but
Yeah,
there will be ways to benchmark. Like I will show. Like there are. It's not like so. There will be some mechanisms to benchmark to say whether the output is good or bad.
yeah, there are ways to see if it is a complete genome, like what constitutes a
Yes,
complete genome.
correct.
Yeah, I wanted to know more about that because it wasn't said anything.
Yes. So that let me come to the like towards the end. I will probably describe
Okay, okay,
how one can measure that.
okay.
How people usually do that, right? So let me
Okay.
come to that later.
Thank you.
Yeah. Okay. So let me talk about this overlap graph. And in this case, what I'm showing you is a visualization of the overlap graph constructed using a small number of reads from human chromosome 20. And in this visualization, basically each colored rectangle is a read, and the black lines indicate the overlaps between those reads. So what you can see here is that the graph is too tangled. That is, it is very hard to say what the actual genome sequence is. So for example, if I start from this node, or let's say I start from this node, purple node, whether I should go next to the green node or whether I should go next to this node, it's not clear, because if you have too many branches in the graph, then it will be difficult to know the exact walk in this graph that I should do to spell the actual genome sequence. So, so in short, I want to say that this graph is too dense or too tangled to work with. There is a need to simplify this graph. And for that, what one technique that genome assemblers use is that they will get rid of all the reads from the graph which are part of the longer reads. So for example, if you see here, R3, read R3 is a substring of read R4. So if read R3 is fully contained in read R4, then it is very likely that read R3 is redundant. And then the question is, why do we keep redundant information in the graph? Why should not we get rid of it and simplify the problem? So this is a common technique that the genome assemblers will use. That is, they will get rid of millions of such reads which are contained in longer reads. So in this case, if R3 is, let's say, fully contained in R4, then the vertex corresponding to read R3 will be removed, and the edges incident to this vertex will also be removed. So in this case, the graph would simplify, right? So in most cases, so the question is this a good heuristic or a bad heuristic? Well, in practice, it works almost well, because if you see, this was the original graph that we were working with. And as soon as you remove the contained reads, you can see like the graph simplifies a lot. It is already starting to give you a linear structure, telling you that maybe you need to start your walk from the left side and you need to go as much as possible towards the right end to spell your original genome sequence. So because the graph is now much sparser, it kind of indicates that this heuristic is actually very useful. But unfortunately, this heuristic is not always safe. So here is a counter example to indicate that this removal of contained reads can create errors as well. To show this counter example, let me draw again a toy example in which I have two copies of the chromosome, one from maternal, one is maternal copy from mother and other is paternal copy from father. And these two copies, let's say, differ at this character, that is the maternal copy is G and paternal copy is T. So now when I sample reads from my unknown genome sequence, let us say I get a few reads as an output from the sequencing machine, and the sequencing machine will not tell me which read came from maternal or which read came from paternal. So that information is not available when the sequencing process happens. Now, in this particular case, observe that the read R6, which is sampled from the paternal sequence, is fully contained in the longer read R2. So this read R2 is the longer read, and read R6 would be a substring of read R2. But now if we remove this read R6, if we construct the corresponding overlap graph, it turns out that there is no read that is bridging an overlap between read R5 and R7. So previously this read R6 was connecting read R5 and R7, but now when we remove it, there is no connection left to spell out the walk from read R5 to R7. So in other words, there is no walk in this graph which spells the paternal sequence. You still have a walk which spells the maternal sequence, but the walk spelling the paternal sequence is lost. So this is a counterexample to indicate that this removal of contained reads is not always safe, and there will occasionally it will lead to errors in the graph construction process. And despite this known limitation, like this, one can argue that, okay, why don't we then not use this heuristic? But it turns out that this heuristic is almost like if you don't use this heuristic, then the graph will be too dense and too impractical. Like the problem will become impractical to solve. So this heuristic is kind of a necessity or important part that the assemblers cannot get rid of right now. So I hope this issue of removal of contained reads is clear. And the project that I displayed initially, in this project, we are trying to address two questions. That is, first is how frequently do such gaps occur in the entire genome sequence? Because if these gaps don't occur in practice, then we don't need to worry about this problem at all. And the second question is, how can we make existing genome assemblers resilient to this problem? So these are the two questions that we wanted to ask in this project, and I'll answer, I'll take you to the answer of these questions one by one.
Dr. Chirag, just one quick, sorry.
Yes,
if you move to the previous slide, for example, let's say R6 is contained in R2. What if we remove R2 and keep R4, R5, R6, and R7? Like remove the bigger one and keep the smaller ones, smaller overlapped ones.
Yes. So in this particular example, maybe that solution works. Yes, but what I'm trying to say that maybe the
there will be cases where this will not work,
yes.
but I can understand that. But what if is there would we get better performance if we keep more overlapped and shorter sequences as compared to removing smaller ones that are contained in bigger ones?
And also, can we just refragment the longer sequences? Will it help?
Yeah. So I think so far I don't to answer the first question. So far we don't know like we don't have a provably good solution to this problem yet, and that will, I would leave it as an open problem in the end. Like these, that is, we don't have an elegant solution that will guarantee uh, this issue, like that will guarantee a good solution to this problem. So let me maybe come to that later. Like, so right now, because if we change this heuristic, either we will end up with a gap, or or the graph will be too dense, is what my experience has been. Like so far, there is, like, we have not found any simple way to get rid of this problem. The second thing is, the second question is, I think maybe what if we bring back this read by matching this, like what if we try to consider each and every read and match here, and see whether the gap closes. Is that the second question? Or the second question is, I think, can you repeat the second question?
I was saying, like, can we just refragment the longer sequences with the help of shorter sequences? I mean, breaking the longer sequences into smaller sequences with the overlap of the shorter sequences. I mean, we'll break the R2 with the help of R6 into three segments, smaller fragments, and we'll remove the fragment which will completely overlap with the R6.
Yeah, sure. I mean, what I'm saying is, this is very, like on the left hand side, I'm showing you a very toy example, right? But in practice, there are millions of leads. So, like, one would need to come up with a general framework of solving this problem. Like, for example, what I'm trying to say is that this is just one toy example, but in practice, we are working with millions of leads. So one has to be like generalize the solution better and see whether on complete data it is working or not. So
As I
yeah.
remember, there exists one algorithm. I mean, oh yeah, one algorithm to address this problem. Are we going to discuss that one only?
So maybe let us discuss, if the time allows. Let us discuss towards the end. Like, is that okay?
Okay.
Yeah. So yeah, okay. So basically, in this case, so the first question is, how frequently do such gaps occur? The second is, can we make the assemblers resilient to this problem? So for the first question, like, let us go to the first question. And for the first question, basically one can ask the following. Like, you can ask the user to give the read length distribution, and you can ask the user to give the number of reads. And then to understand what is the frequency of a gap, what I can do is I can consider all possible sequencing outputs that satisfy this constraint. That is, the number of reads should be fixed, and you are given a certain read length distribution. So you can consider all possible sequencing outputs, and then among them you can check which of them lead to a gap. But the problem here with the calculation of this formula is that if we consider all millions of reads, and each read can be sampled from any of the billion characters on the genome, so this leads to exponential number of possibilities in which sequencing can happen. So checking all of these exponential number of sequencing outputs will be computationally prohibitive. In this work, what we did to get around this problem is that we said that, okay, can we characterize the exact conditions when these gaps occur? And for that, we gave out a claim in the paper that is, like, to characterize exactly in what conditions do such gaps occur. So that is, if you consider the reads which cover the mutation position, like this is the position where mutation is present between paternal and maternal genomes. And if we consider X1 as the read covering this position and which extends farthest to the right. Similarly, the read on paternal copy, which extends farthest to the right, it is extending up to X2, and this read is extending up to position X1. So based on these two values, we can give out the exact conditions that these reads need to satisfy for a gap to occur. So for example, simple condition is that X1 should not be equal to X2. There should be at least one read in this interval, and so on. Like, I'll leave out the details, but these are like conditions that need to hold for this gap to occur. And the reason these conditions are helpful now is because if once these conditions are true, we know that the gaps will occur. And due to these conditions, what we can do is that we can calculate this formula more efficiently. So we can compute this ratio in polynomial time using generating functions and inclusion exclusion principle. So these are the principles like which allow you to measure like in combinatorics. Uh, you can like combinatorially uh, define the space, and you can count how many such inputs will satisfy this given property. So in polynomial time, we compute this fraction using these conditions, and we then go and look at different read-length distributions produced by different sequencing technologies. So for example, on the x-axis, you see the genome sequencing depth, that is how much data that you're generating. And on the y-axis is the frequency of at what frequency will you see the gap. So what I'm showing you here is that this frequency of gap will change from sequencing technology to other. And the reason why different sequencing technologies will have different gap frequencies is because they have different read length distributions. So imagine if there's a sequencing technologies which produces reads of exactly the same length, then you won't see any gap, because if reads are of exactly same length, then it will never happen that a longer read is contained in a shorter read. So it turns out that for example, in PacBio hi-fi case, the gaps frequency is much lower. And the reason is exactly, this is why, that because the PacBio hi-fi, the read lengths are in very narrow range. Whereas in other technologies, the reads lengths tend to be very variable. So you can have highly variable read lengths for other technologies, and there the gaps are more likely to occur. So with this question, now we know that the gaps can occur, and especially when you are using sequencing technology which produces reads of highly variable lengths. So now the question is coming to the second question: how can we make existing genome assemblers resilient to this problem? So for this question, maybe I think the first thing that comes to mind, and I already gave a hint, is that if all the reads are of equal length, then this problem will not occur, right? So that is the one simple solution that probably comes to mind is, okay, why can't I just fragment all the reads into shorter reads of equal length? And that can be done by just 2 lines of Python code. That is, you take all the reads and chop each read into equal length fragment. So what is the problem with this solution? Well, the problem with this solution is that, remember that you have repeats in the genome sequence, and the reason why we would always prefer longer reads over shorter reads is that longer reads can bridge over these repetitive regions. So if you have a repeat, then this longer read can tell you how to assemble this part of the genome, and this longer read can tell you how to assemble this part of the genome without any ambiguity. So now if you fragment all the reads, then that information is lost. So we won't be able to assemble these repetitive regions of the genome. So how do we address this? Well, the heuristic that we gave in the paper was to do something called repeat aware fragmentation. That is, what we do is we detect for every read, we predict whether it is spanning the repetitive region of the genome or not. And how do we do that? Well, one way that we follow is we check overlap of read with all other reads. So this is a visual of that idea. That is, if you have a read A, which is coming from a non-repetitive region of the genome, then some number of reads will overlap with that read, right? Because the reads which are sampled from similar region will overlap with that read. But on the other hand, if a read is sampled from a repetitive region, you will see a larger number of reads overlapping with that read. And by looking at the number of reads that overlap with my current read, I can try to predict what regions of my read may be repetitive in the genome sequence. So for example, in this read B case, if I see a large enough number of reads overlapping here, then I can predict or heuristically sort of infer that this region may be repetitive and I should not do any fragmentation in this region. So in this repeat fragmentation idea, basically we will do fragmentation in a naive manner for reads like read A, but in read B cases, we would avoid the fragmentation when we see a repetitive region in the read. So if you have a repeat, then we would want to preserve the entire read length in that region. And this is the idea that we give so that we don't lose the ability to assemble repetitive regions of the genome. So let us see how this idea does in practice. Okay, so let me also show you one thing. So let us say, so this plot is showing you the read length distribution before and after fragmentation. So before, you see this black histogram indicating a wide read length distribution. That is, you have reads lengths ranging from a few thousand bases all the way to 200,000 bases. But after repeat fragmentation, now the read length distribution has narrowed down, and that's why we will see that this heuristic will help in avoiding those gaps in practice. So now comes the benchmarking. So to benchmark this idea, we basically integrated this idea in an existing assembler, which is the state-of-the-art assembler. So this is known as IISm. So we call our method as repeat-aware fragmentation tool. So we compare IISm with Raft IISm on multiple data sets. And actually this table is very dense because for benchmarking, usually we have to measure a lot of properties. That is, what is the length of the assembled sequences? Are they accurate or not? And so on. But let me point you to the two main metrics. That is the average length of assembled sequences and what are the number of sequences that were assembled end to end. That is, what is the number of chromosomes that were assembled completely. In both cases, you can see that the contiguity of the assembly is improving when we do repeat-aware fragmentation. So there are two numbers here because one is for paternal haplotype, that is father, because you have two copies of the genome, one from father and one from mother. We are measuring them separately. And for both copies, you can see that there are improvements in the length of assembled sequences and also the number of gapless chromosomes assembled end to end. One can also see this visually. So it may be the better way to see this is to do a visual interpretation. So on the left-hand side is the IISm assembly on dataset B4. Each white pixel or white rectangle that you see is a break in the assembly in this picture. The pink corresponds to one haplotype and the blue corresponds to paternal haplotype. And you can say that you can already see that on the left-hand side, you can have too many breaks in the assembly, whereas on the right-hand side, there are fewer breaks. So this is to indicate that the contiguity of the assembly has significantly improved by this repeat fragmentation idea. So let me now summarize and I'll take more questions after this summary. So basically, what I wanted to show you is that the routine generation of complete gapless human genome is important for biological applications. And there's a long road forward. It requires further innovation in assembly algorithms. The particular topic that I addressed in my talk, I addressed that existing assembly algorithms are suboptimal when there is significant variability in read lengths. And to address that, we can do a repeat-aware fragmentation of those reads. And using that, we see improvements in the assembly quality in practice. Well, so then I leave this question for all the students. That is, so the solution that we gave is a heuristic. That is, we cannot guarantee we cannot do any guarantees that all the gaps will be resolved or maybe 90% of the gaps will be resolved. We are not putting any guarantee like that, but is there any provably good algorithm to solve this problem? That is a question. So I leave that as an open question for the students to think about. And yeah, so I'll just close by acknowledging all the lab members and students, Sudanwa and Mehak, who led the project, and Professor Devnath Pal, with whom this project was done in collaboration. So I'll close here, and I'm happy to take more questions. Thank you.
Sir, I